Hello, welcome to 13 Universal. This is A-level CIE biology, and I want to say at this point that all the questions we're using here is from CIE past papers I'm asking. Alright, so we are on mitotic cell cycle, MCQ paper 1 series 5. So we wish you the best as you write your final exam, okay, in about a week's time. Okay, so this is Shedro, your teacher. So let's go on to the question. Bring your your writing materials. Let's crack some questions. Let's fry some fish, okay, and make fish pie. All right, come on. See the first question there. It says the enzyme, the enzyme telomerase prevent loss of telomeres. Okay, that means it replenishes telomeres after many mitotic cell cycle. So it keeps replenishing it. So let's see which cells need to transcribe telomerase enzyme. Which cells? Cancer cells, stem cells, activated memory lymphocytes. Okay, so which of them needs to transcribe telomeres? So which of them does not prevent loss of telomeres? So pause this video and try to solve this question by yourself. Try it by yourself. Okay, remember that telomeres are usually found at the tip of chromosomes. Okay, and they consist of repeated base sequence that prevent loss of genes. Okay, so as a cell DNA replication occurs during cell division, so they're usually found at the tip, right? So telomeres, right, are found at the tip here. So, and they consist of repeated base sequence. Their job is to prevent loss of gene or loss of genetic information. Remember that normally, as cells divide, so we have cell division. Cell division, I'll call it CD, cell division. As cell divides in normal cells, okay, the length of telomeres depletes or reduces. So cell division is actually directly proportional, right, to the length of telomere, LT. So you see that both of them are proportional to each other. So as the cell division increases, all right, what happens is that the length of telomere of course decreases. I think that's in direct proportion. So as cell division increases, what happens to length of telomere? It decreases. So that means that cell continues to divide until it comes to a point where all the telomeres are depleted and the cell dies, right? So we can as well say in terms of lifespan, right, that length of telomere, right, is directly proportional to the lifespan of the cell. Okay, so in normal cells, telomeres are not replenished, they are not lost. Okay, but there are cells that must not die. Remember that as cells divide, length of telomere depletes, and then at a particular point when it is fully depleted, telomeres are depleted at the tip of the chromosome, the cell will die, and that is what we call apoptosis. Have you heard about apoptosis? Apoptosis is programmed cell death. Apoptosis. Okay, now all cells are programmed to die. All cells. This is the science, the chemistry, or the biology behind aging. Okay, so cells age. Why do they age and die? Because as cells divide, what happens is that telomeres, telomeres are depleted, and it comes to a point where the telomeres are completely depleted. So genes are lost, especially genes that the cells need to survive. So the cell dies. That's apoptosis, programmed cell death. So all cells, there are cells that should live longer, right? So these cells that undergo this kind of apoptosis, the telomerase enzyme is usually inactivated. Okay, but there are certain cells where it is activated. Telomerase enzyme is an enzyme that prevent loss of telomeres so that the cell can continue to divide indefinitely, guys. If we can find a way to make telomerase enzyme to work in normal cells, that means we're not going to age. That's the elixir of eternal youth. We're not going to age. We live younger. Okay, so we want to live younger forever. That's the elixir of eternal youth. But unfortunately, normal cells have their telomerase enzymes inactivated. Therefore, our cells continues, right? The telomerase depletes, okay, and the cells undergo apoptosis. That's why the older people shrink. Cells die, they're not replaced, okay, and they form wrinkles on their faces. All right, let's come here. So, which cells need to transcribe telomerase enzyme? Cancer cells, sure. Cancer cells. Remember that cancer cells, they divide indefinitely. So their telomerase enzyme, all right, is active. So they continue to divide. So this needs it. Stem cells stay long in the cells because they are the ones that make other cells, supply other cells. So they need, right, to transcribe the telomerase enzyme. Activated memory cells. Now memory cells are very important because they help to protect the body. They give the body active immunity, and they always allow rapid response to rapid immune response to a pathogen that has been in the body before, okay, in further invasion of a particular pathogen. So 3 of them need to stay longer. So their telomerase enzymes need to be transcribed so that their telomeres do not suffer loss, and then they stay longer. So the longer the cell needs to stay, okay, the more needful or necessary for telomerase enzyme to be transcribed. So these 3 cells, all right, need to stay. Of course, they stay longer, they live longer. Cancer cells are immortal, they don't die. Stem cells need to stay longer in the cell because they supply other cells, and activated memory cells also need to stay longer to provide immunity, keep immunity. So the answer is A. Is that okay? All right, let's run. Which processes that occur in the cell cycle are represented in the diagram? Which of them, the one that occur in the cell cycle? Yeah, so guys, pause the video and show us what you got. Your exam is next week, 11th of June, 2024. You ready? So try this question. Don't check the masking, okay? So let's see what happens. Now I'm going to use something else to explain this. Remember that in cell cycle, guys, cell cycle is like this. Pardon my diagram. See, now this is mitosis. All those prophase, metaphase, anaphase are inside there. This is cytokinesis, remember? Okay, see. The rest is interphase. Okay, the rest here, the rest here, the interphase. But the first part of interphase is, okay, S1. Is that okay? No, don't call it S1. Call it G1. Yeah, G, growth phase 1. G1. Yeah. And the second part of interphase is S. That's S here, synthesis phase, and this is G2. Okay, you see, so in G1, the cell is young. It's making protein synthesis occurring here, okay, making new organelles. At S phase, it doubles its genetic material. G2 makes central checkpoint, checking whether DNA has genetic errors, okay, and then more ATP. And that, okay, in mitosis, the nucleus divides, and in cytokinesis, the cell split. So the cell starts out like this, guys. See this young cell, very promising in life, very young, okay. This cell is young, and people meet this cell and they ask this cell, "Hi, young chap, what do you want to be?" Do you know the answer the young cell gives to people that meet it? It says, "I want to be like mama. I want to be like my mom." That's what it means. So what do you mean by the fact that you want to be like your mom? So I want to grow up and split. Okay, so okay, don't worry. So this cell actually, undergoes the process, grows bigger like the mother cell. You can see it's doubling the genetic material here. Now can you see? So it grows bigger, doubles genetic material. Okay, yeah, and then the next thing it does is that watch it. So the cell says, okay, I've doubled my genetic material. I'm thinking about the daughter cells, my daughters. So I'm going to divide the genetic material, okay, into two. So it splits the nucleus, right? That is mitosis. Is that okay? So mitosis, can you see the same cell? All right, that's mitosis. Mitosis, you can see that, okay? So you can see now. You see, now this nucleus is split, right? The cell said, okay, it's time to actually let the entire cell split so that I can have my daughter cells, my babies. So the cell undergoes cytokinesis. Can you see? Invagination. This is animal cell, right? So that's it, guys. Now watch this. All right? So the cell splits into two. Don't forget this, because I'm going to use it to explain a lot of things in the future. All right? So of course, by the time it splits, it gives rise to this young cell, and it continues. Of course, it's going to give us two of them, right? Each of them enters the cell cycle, continues the cell cycle, all right? And it never ends until the telomeres are split, okay, depleted. Telomeres are depleted, and then the cell dies. All right, look at it. Now can you see the mass of, they said, which processes that occur in the cell cycle are represented in the diagram, right? This is the mass of nuclear DNA in a cell. This is time. Now, can you see the first option says DNA replication occurs. Okay? Yeah, of course, DNA replication is occurring. How do I know? Because the amount of DNA increases. Right? So during DNA replication, there is an increase. Right? DNA is doubled. That's what is happening here. Can you see? Yeah, the genetic material doubles here. So it occurs here. Then as a nuclear division, nuclear division occurs because during nuclear division, can you see nuclear division? You can see, can you see what's happening here? The nucleus splits into two. Okay, the same cell. So nuclear division occurs here. Okay, it occurs here because we still maintain the same double DNA. Okay, 4 arbitrary units maintained. Now, but that's not all. There's this part of this graph, can you see, where DNA decreases, and that is cytokinesis, right? So can you see, now when the cell undergoes cytokinesis, is split. This is in one cell, one big cell split into two. So when it splits, now each cell has the original mass of DNA, which is 1 according to this, okay? So it's going to involve DNA replication, nuclear division, and cytokinesis. So that should be B. So all of these, right? Of course, this is nuclear division is mitosis. Okay, cytokinesis. All of this is interphase, right? Okay. So during interphase, we have growth, nuclear division. Now this is DNA replication, which is part of interphase. Okay, but the cell has to grow. Okay, that's cell growth phase 2 also. So this shows the entire cell cycle. The entire cell cycle includes DNA replication, nuclear division, and cytokinesis, and then cell growth anyway. So B is the answer. All right. So let's go to the next question. A gene code for the production of protein P53 that binds to, look at the function of this P53. It binds to damaged DNA during, right, of course, interphase, and prevents its replication. So P53 does not allow replication of damaged DNA. That's the function. Now, a carcinogen in cigarette smoke mutates the gene coding for P53. So the gene that codes for this protein is mutated, all right, preventing production of the protein. So the protein is no longer produced. Which statement explains why this mutation may cause cancer, guys? Okay. Now I want to pause the video and try to answer it. Why does this mutation, why do you think it might cause cancer? May cause cancer. Right? Pause the video and try it. Okay, look at A. cells with no p53 are able to undergo mitosis. Yeah, they're able to undergo mitosis, but it doesn't explain why it may cause cancer. Okay, remember, the function of this p53 is that it prevents replication of damaged DNA. So if the DNA is not damaged, okay, the cell undergoes mitosis. But you see, it does not prevent mitosis. What it prevents, all right, is the replication of damaged DNA. Okay, so A does not explain that. All right, so let's go on. Just try to eliminate and get what we need. So A is out of the way. Okay, now B says cells with no p53 replicate their damaged DNA. Yeah, they replicate their damaged DNA, and that is a problem. So if they replicate their damaged DNA, because p53 prevents replication of damaged DNA, it binds to damaged DNA, right, and prevents replication. So if damaged DNA replicates, it could result to cancer. So that's correct. So why is C wrong? Says the carcinogen in cigarette smoke increases the rate of cell division. Well, that is not correct. Okay, so it doesn't increase cell division. It might cause cancer, but doesn't necessarily mean. Of course, cancer will result, all right, in the increase in the cell division, all right, but not all cell division results to cancer, right, except a malignant tumor is formed. Okay, so this says the mutation p53 causes uncontrolled cell division. Uncontrolled cell division may occur, right, but does it result to cancer? Right, but how does it? The reason why it could cause cancer is because of p53, right? Is no longer working. So if p53 is no longer working, this is the only reason. It does not actually, okay, result. If it mutates and p53 is still produced, that means the DNA, damaged DNA will not be replicated, and we cannot have this uncontrolled mitosis. Okay. All right. So, but remember, guys, what this does is that it prevents the replication of damaged DNA. All right. But for us to have uncontrolled mitosis, that means that there's a mutation on the oncogene and the tumor suppressor genes. Is that okay? The oncogenes, proto-oncogenes. Proto-oncogenes mutate to form oncogenes, and then proto-oncogenes regulate cell cycle. So that is actually the one that will determine this uncontrolled cell division, right? Because when proto-oncogenes becomes oncogene, it causes the cell to divide. The cell continues to divide because oncogene does not respond to signals to stop cell from splitting when it's not meant to split. All right, so this does not deal with p53. Okay, so let's go to the next question. So the answer should be bravo. All right, yeah, let's go on quick. Now you can see that stained onion cells undergoing mitosis were observed using a microscope. Which role is correct for mitosis in plant cells? Which role is correct? Okay, let's check it out. Pause the video and check it out. Which role is correct? Let's check role A. During prophase central visible, okay, metaphase chromosomes pair up at equator. Chromosomes do not pair during mitosis. They pair in meiosis, and in meiosis they pair in prophase, not even metaphase. Okay, so that is why A is wrong because of this, right? You can see because of these statements that makes A wrong. So this is wrong. Now if you come to B, centromere present, okay, chromosomes align at equator. That's correct. Yeah, so this is correct. That is correct. Chromosomes replicate to form chromatids. Chromosomes do not replicate in anaphase. Sister chromatids separate. All right, see, each chromosome is visible as two chromatids. Yes, this makes sense. Centromeres attach to spindle fibers. Yes, that happens in metaphase. Chromatids separate and migrate to opposite pole. That happens. Chromosomes decondense. That happens. So everything in C is actually correct. Alright, this is wrong because of, alright, the anaphase part. A is wrong because of the anaphase part. Now look at D. Spindle fibers formed by centriole. Yes, that's correct. Centromeres attach spindle fiber. Yes. Chromatids are pulled apart by centriole. Okay. Yes. Anyway, the microtubules formed by centriole. Spindle fibers form nuclear membranes. Spindle fibers do not form nuclear membrane. Okay, so that disqualifies D. Alright, so the answer is C. So it says 3 parts of chromosomes. Okay, 3 parts of chromosomes, let's look at it. And their functions are listed. Okay, so 3 parts and their functions are listed. Then the question says, which part is matched with its correct function? Okay. Alright, so I want you to try this. You're ready for the exam? Try this. So let's check A. P1 is centromere. F2, centromere, let's check it, holds 2 sister chromatids. That's correct. So we just got the answer in the first place. Okay, but we also have to tell why the answer is, why other answers are wrong. So you can see this. Okay, these are 2 sister chromatids joined by centromere. So what is in the middle here is centromere. This is centromere. Okay, yeah. So A is correct. But if we check B, it said P2 histone proteins goes with F2, holds 2 sister chromatids. Nope. So that is wrong. Now if you check C3, it says histone proteins prevent loss of gene. Nope. What prevents loss of gene is centromere. Telomere. Alright. The P3, which is telomeres, holds the coil of DNA together. Now that is a function of histone protein. Okay. Of course, this is wrong. So the answer is A. Quickly, which features of mitosis, right? Which features of mitosis are important for a single-celled organism? Single cell, one cell organism. Okay. Now, which one is correct? Asexual reproduction. Yes, mitosis is the basis for asexual reproduction. One parent, okay, divides by mitosis, correct? Now, production of genetically identical cells. Yes, mitosis helps to produce genetically identical cells. Growth. Yes, mitosis helps in growth, but in multicellular, because as cells continue to increase in number, the multicellular organism grows. But in single cell, mitosis does not help in the growth of single cells. What helps is protein synthesis, okay, and increase in number of organelles, okay, ATP synthesis and all of that. So 3 is not correct. So it's only 1 and 2. Okay, so the answer is B. Consider. All right, some events in the cell cycle are listed. Which events occur in mitosis? So which of them occur in mitosis? Pause the video and try it. Which ones occur in mitosis, guys? Right? Remember, in mitosis, if you remember, yeah, this is cell cycle. Sorry for that. Cell cycle. You can see this. Yeah, this part is mitosis. Okay. Yeah, that's mitosis. All right. But that part is cytokinesis. So mitosis is this part. So the acronym for mitosis is PMAT. P is prophase, PMAT. M is anaphase, right? T is telophase. Okay. So interphase is this part, right? This is interphase, which is made up of right? Of course, let's start from here. G1, growth phase 1, where the cell grows bigger, makes protein. Okay, part of it is S, where DNA replicates semi-conservatively, and we have G2, okay, where DNA is checked, right, and central replicate. So you see, the only part of mitosis here is not interphase, is not part of it, is metaphase. Cytokinesis is cell split into two, so the answer is 2 only. All right, let's go, guys. Hope you are warming up. During the cell cycle, the mass of DNA in a cell is doubled. Yeah. At which stage in the cell cycle is the mass of DNA doubled? Is of a cell in at which stage in the cell cycle is the mass of DNA in a cell reduced? Where is it reduced? Okay. Yeah. Pause the video and try it. At which stage is it reduced? Okay. All right. So it's not reduced in anaphase. Is reduced in cytokinesis. Okay. In fact, in interphase it is doubled. The S phase of interphase doubles it. Nothing happens to it in prophase. Okay, the same, because it's doubled in interphase, right? Then it's reduced in cytokinesis, right? So cytokinesis is that stage B where it is reduced. How many of the listed structures typically contain genetically material that has telomeres? Yeah, telomeres. Okay, telomeres. What is telomere? So how many of these? Okay, telomere is found at the tip of chromatids, right, of chromosomes that prevent loss of gene, right? So they are found at the tip of chromosomes, right, chromatids, right? Bacterial cell. Pause the video and try it. Bacterial cells, do they have chromosomes? They don't have chromosomes. What they have is DNA. So chromosome must contain DNA and histone proteins. Okay, so bacterial cells, their genetic material lacks, right, histone protein. So we can't call it a chromosome. So it doesn't have telomeres. Okay, it doesn't have it. Okay, yeah, sorry. So bacterial cell doesn't have telomeres. Okay, chloroplast has DNA, but it doesn't have histone protein. Therefore, it doesn't have telomeres. Telomeres can only be found where there's chromosome. Chromosome consists of DNA and histone protein. So whatever doesn't have histone protein doesn't have telomeres. All right. Mitochondria has circular DNA just like chloroplast, but there's no histone protein, so there's no telomeres. Nucleus contains chromosomes because it's made up of the chromatin there that forms the chromosome consists of histone protein, all right, and DNA, so it has telomeres, all right. So it's only one structure, which is nucleus. So the answer is A. All right, guys, hope you are with me. Let's run. Yeah, in eukaryotes, the chromosomes become shorter and thicker during mitosis. Yeah, the thickening may be increased by molecules called protamines. The protamines replace part of the structure of the chromosome. Okay, which part of the chromosomes is replaced by protamines? Remember, the protamines, what is their function? They help in the thickening. It's increased. So they say the thickening or condensation is increased by molecules called protamines. So which of them help, right, in the condensation or thickening or, all right, folding of chromosomes? Which of them? Is this centromere? Pause the video, all right, and deal with it. Fry this fish. I am hungry. Okay, is it centromere? Centromere, is it involved in thickening, right, or folding, okay, of chromosome? Nope. Centromere holds the sister chromatids together. You know it. So this one I'm drawing now, this circle that's holding the sister chromatids, that's centromere. So centromere doesn't hold. Chromatids are this single-stranded genetic material held together. So that is not what helps in the folding of chromosome. Yeah, histone protein. Yes, histone protein helps in the folding. Okay, histone protein. Remember, we have 8 histone proteins against one um, molecule of DNA. So we have 8 of them, right? Okay, you have 8. Is that 8? Yeah. Then the molecule of DNA continues to fold around it and then condense. Right. So that's correct. Telomeres, nope. Telomeres are found at the tip of chromosomes like this. Okay. All right. This is telomere. They prevent loss of genetic information, loss of genes. Okay. They're the ones that determine the lifespan of a cell. The longer the telomere, all right, the longer the lifespan. Okay. So therefore, what are you saying? The answer is C. All right. During telophase, hope you are counting how many you have gotten. During telophase of mitosis, a scientist stains the chromosomes of a diploid animal cell with a fluorescent dye to observe the telomeres. Yeah, I want to see telomeres. This cell has 22 chromosomes. How many telomeres will the scientist observe in one of the nuclei? Okay, that has 22 chromosomes. All right, so how many? Don't forget, guys, you should pause this video and try this. Practice this, right, question. Do not wait for me to tell you the answer or go check the answer. No, remember that a chromosome, look at it. Oh, this is too big. Okay, let me draw another one. A chromosome consists of 2 sister chromatids, right? Now these are 2 sister chromatids which are genetically identical, joined by this. Now this is centromere. Centromere joins these 2 sister chromatids. Okay, and a chromosome contains at the tip of chromosome you have telomeres. So each chromatid has 2 telomeres because it has 2 ends. There's a telomere there. So a chromosome has 4. One chromosome, okay, has 4 telomeres. Okay, yeah. So if one chromosome has 4 telomeres, 22 chromosomes will give us how many telomeres? So 22 chromosomes, which is C, will give us how many telomeres? You can see one chromosome. This is one chromosome. Okay, one chromosome. It has how many telomeres? 1, 2, 3, 4. So 22 will give us how many? You just cross and multiply 22 times 4 over 1. So multiply it by 4. 22 times 4 will give you what? 88, right? So that's going to be 88 telomeres. That is the answer, guys. No, not 88, 44. Sorry. 4 times 2 is 8. What? It's supposed to be, yeah. Okay, look at it. Let's look at it again. So how many telomeres will the scientists observe in one nuclei? Okay? Yeah. So guys, they say during telophase of mitosis, a scientist stains the chromosomes of a diploid animal. That's diploid, okay? Cells with a fluorescent dye to observe the telomeres. Okay? This cell has 22 chromosomes. Okay? That this cell at telophase. Okay? It has 22 chromosomes. Remember that if the cell has 22 chromosomes at telophase, watch you. The telophase already has, yes, it already has 2 nuclei. Okay, so the entire chromosome in the entire cell is 22. That means each nuclei should have 11, right? Yes, so this should be 11 chromosome. Okay, 11 chromosome, right? So the entire chromosome in the entire cell is 22. Okay, that's this 22 here. Is that okay? So they now said, how many telomeres will the scientists observe in one of the nuclei? So in one nuclei here, we'll have 11. Yes. So that means that if one chromosome gives us 4 telomeres, 11 chromosome will give us what? Okay. 11 times 4. That will be 11 times 4. Yeah. 11 times 4, okay, is 44. Is that okay? Yeah. Because the 2 nuclei gives us 22. And we're looking at what is in 1 nuclei. Is that okay? So that's 11. That is why the answer is 44. Is that okay? All right. Yeah. Methotrexate is a drug used to treat cancer. Yeah, guys, let's look at it. Methotrexate is a drug used to treat cancer. It can act as an enzyme inhibitor, preventing synthesis. Look at it, preventing synthesis of nucleotides containing thymine. So it prevents the synthesis of the nucleotide that contains this base, thymine. Thymine is a pyrimidine, okay, found in DNA. But that particular drug, all right, is used to treat cancer and is an inhibitor and prevents the synthesis of this thymine. All right, let's see. Of course, says cell treated with methotrexate are not able to complete the cell cycle. A cell entering telophase is treated with methotrexate. Which stage of the cell cycle will be affected by the action of methotrexate? Okay, so the question I want to ask you is, which stage requires, all right, that particular, don't forget that which stage uses the nucleotide, which stage uses nucleotide. So pause the video. I want you to think through it. Which stage uses nucleotide? All right, that has thymine. So what is nucleotide? Which stage uses it? Which one? Pause the video and try it. So the stage that uses nucleotide is the stage that builds DNA. Nucleotide is a monomer for DNA. Okay? So which stage synthesizes DNA? Semi-conservative replication of DNA. Which stage, all right, helps to synthesize DNA? Of course, that's the S phase of interphase. All right, anaphase does not build DNA. Metaphase, at anaphase, the DNA, anaphase, the chromatids, sister chromatids separate, they move to the opposite poles. Metaphase, they align at the equator, they line up. All right, so DNA is not built here. Prophase, they coil up, all right, condense. So the only stage where we have this is the S phase of interphase. That is where nucleotides are actually needed to make DNA. So if during that at anaphase nucleotide containing thymine is not present, so that means that DNA synthesis cannot occur. So it affects the cell cycle. And if DNA is not synthesized, other part of the cell cycle will not occur. So B is the answer. Interphase. That is where DNA is actually built by semi-conservative replication of DNA at S phase of interphase. All right. Yeah. Which stage of mitosis is correctly described? So which stage is correctly described? So I want you to check it out. Okay. Which stage is correctly described? I've given you clues. Pause the video and solve this question. In anaphase, chromosomes line up across the equator. Is this correct? Anaphase chromosomes, do they line up at across the equator? Nope. So this is describing metaphase, not anaphase. Okay. Now B says in metaphase the central replicates. No, that is G2 of interphase. And of course, in prophase, chromatids move to the opposite pole. Nope, this describes anaphase. C describes anaphase. Then D must be the answer. In telophase, chromosomes uncoil to form chromatin. Yes. All right, they uncoil. Remember that telophase, part of what happens in telophase is the opposite of what happens in prophase. So in prophase, they coil up, but in telophase, they uncoil. So the answer is delta. Come on, let's look at this. So, which role correctly describes part of chromosomes structure? Yeah, present during mitosis. Okay, correctly describes Okay, pause the video and try it during mitosis. So region of centromere, let's look at A. Region of our chromosome. Okay, let me get the pen. Let's tick it out. Region of chromosomes with no DNA. Okay, it has DNA. Okay, but non-coding. All right. So this is not quite correct. Okay. Now chromatin says single DNA polynucleotide with histone protein. No, it has double, right? DNA is double stranded, so this is wrong. So we don't need to continue. Let's go to B. Region of non-coding DNA, okay, holding two chromatids together. Yes, this makes sense. Double-stranded DNA molecule with histone protein. This makes sense. Okay, telomeres, region of DNA with many short repeats of sequence of bases. This makes sense. Okay, yeah, the answer looks like it's going to be, but let's see. C. Region of DNA with no histone protein that allows separation of chromatin. That allows separation, okay? Yeah. DNA molecule coiled around histone protein to form a chain of nucleus. Okay. There's a region of DNA with no protein that protects the end of a chromatid. So what is wrong about C? Okay. Telomere says region of DNA with no protein that protects, right? All region of DNA, they have protein, okay? Because DNA consists of, I said, region of DNA with no proteins that protects the end of chromatin. No. This is not region of DNA. It's region of, telomere is the tip of chromosomes, okay? That protects, all right, genes. So this is wrong, all right? So this is wrong. Of course, we have established that this is wrong and that this is not right. Let's look at the region that attaches to spindle microtubules, okay? Microtubules and divides. It doesn't divide at prophase. Centromere divides, okay, during anaphase, right? Yeah, it divides during anaphase so that sister chromatids can separate, right? It divides anaphase. So this is wrong, okay? And that makes this wrong. So the answer is B, guys. The diagram shows relative time taken. Let's roll. For each stage in the cell cycle. All right. Yeah. So the question says, which role identifies correct sequence of the stages in cell cycle? So, all right, which stage? Don't forget that the biggest part is interphase. Z is interphase, all right? All right, now I want you to pause the video. I've given you a clue. Solve the rest. What's the answer? All right? And then, of course, the next bigger part is mitosis, right? And then this is cytokinesis. So C is cytokinesis. Look at the answer here. All right? Y is interphase and X is mitosis. But sometimes this can also be divided into three parts, which is G1, right? S and G2. Okay, so the answer is C. All right, let's run. Which description of the chromosome structure is correct? Which description of chromosome structure is correct? Okay, so let's see. So pause the video and carefully think through it. Try to solve this question. Now, which description of chromosome is correct? Now A says a chromosome at the beginning of interphase consists of two identical chromatids. At the beginning of interphase, okay, at the beginning of interphase is G2, sorry, G1. So at G1, DNA has not been doubled, so we cannot have two identical chromatids, all right, because S phase has not occurred. Okay, remember that at interphase the first is G1 and then S phase. So at it's during S phase that DNA, all right, semi-conservative replication occurs. So at beginning G1, you can't have this. So this is why A is okay. I think is wrong. Yes. B says telomeres are length of non-coding DNA. Yes, found at one end of each chromatid. They are not found at one end. Telomeres are found at both ends, not one end. Okay. Of course, they prevent loss of gene, but they are found at both ends. So all right. So if this is a chromatid, telomeres are found at both ends, here and here. Is that okay? So that is why B is not correct. C says the two chromatids, we're describing chromosome, said the two identical chromatids of a chromosomes are held together by a centromere in which there is no DNA. Okay, there is DNA in chromatid. Is that okay? In centromere. It says they are held together by a centromere, and they say that centromere has no DNA. Centromere has DNA. Okay, so this is C is wrong. So we are left with D. D should be correct. So the very long molecule of each chromatid, okay, is called and held together by protein called histone. Yeah, histone holds it together. DNA, right, held by histone. All right, which processes involves mitosis? Yeah, come on. So which process involves mitosis? Now, clonal selection, right, of forming of cells at the root tip of a growing root, replacing skin cells damaged by an injury. Which one? Pause the video and solve it. Pause the video and try the question. Okay. So now, yes, of course, clonal selection does not involve mitosis. Okay, it involves antigen recognizing, all right, the appropriate receptor on the T lymphocyte. So T lymphocytes have receptors, so the receptors should be able to fit. If the receptor fits into the antigen or the pathogen, right, that's close. So it doesn't involve mitosis. Yeah, forming of cells, anything has to do with forming of cells involves, all right, at the root tip, that's mitosis. Replacement of cells, also mitosis. So it involves two of them. So the answer should be C. Okay, let's run. A drug has been developed to treat cancer cells. Okay, certain types of cancer. It prevents mitosis by binding. See what it binds to the spindle. It binds to the spindle. Okay, and prevents sister chromatids being separated. So at what stage of mitosis does sister chromatids separate? That's it. I'm moving to opposite poles of the cell. So that should be in your mind. At what stage? The photomicrograph shows cells in different phases of mitosis. Okay. All right. Then I said, what represent the stages of mitosis that will be able to occur in a cell which is entering prophase when treated with this drug? So which one represents it? Guys, don't forget that sister chromatids. All right, pause the video. Pause it. That's the drill. Pause the video and try it. Preventing sister chromatids being separated. Now, don't forget that sister chromatids separate at anaphase. All right. So where is anaphase here? Okay. So where is anaphase? So anaphase is 3. Can you see sister chromatids are separated? They separate at anaphase here. So 3 is anaphase. Okay, yeah, and then 4 is looks like telophase. Okay, no, 4 is 4 looks like interphase. It's not telophase. Okay, that's after cytokinesis. Can see that new cell is formed. Okay, yeah. Now 2 is of course metaphase. How do I know? The chromosomes align at the equator. Now 1 is prophase because okay, because it's condensing. Now but this this 4 also looks like telophase, okay, but it's just interphase. Okay, the cells are new, kind of, you know. You can see, we see how this looks like the spindle fiber. No, not the spindle fiber, the nucleolus. All right, okay. Now, of course, um, so if it prevents sister chromatids from being separated, that means anaphase doesn't occur. So the question says, what represent the stages of mitosis that will be able to occur in a cell which is entering prophase when treated with the drugs? Is only 1 and 2 that can occur. 3 can't occur. And if 3 cannot occur, 4 can't occur. Is that okay? It's only 1 and 2. So once the chromosomes align at the equator, they cannot, sister chromatids cannot separate because the drug prevents it. Therefore, anaphase cannot happen. So it's 1 and 2. Which features of stem cells enables them to replace cells in tissue such as skin? So which tissue, which features? Which feature? Now pause the video and try it. So you can see that there are undifferentiated cells that are present at birth. Now this is not why they will be able to replace cells. Okay, yeah, they are undifferentiated cells. They differentiate to form skin cells. So this is not a major issue. Because if they differentiate to form skin cells, that means that will be the end of stem cells. But guess what, guys? They divide by mitosis. Stem cells divide by mitosis continuously to supply some cells that can differentiate. Yes. Okay, so C is the answer. Right? Now this says they have the full number of chromosomes. No, it's this C that's the feature. Alright, let's run. During prophase, a scientist stains the chromosomes. See this question again being repeated, of a diploid animal cell with fluorescent dye to stain telomere. Now this is during prophase. The other one was telophase. Okay, this cell has 32 chromosomes. So yeah, it has only one nucleus. Okay, right. So how many telomeres will the scientists observe? So I want you to pause the video and then work it out. I've showed you how to work it out. Work it out again. Don't forget, pause the video and work it out. Okay, don't forget that this is a chromosome. This is how chromosome appears at prophase. Can you see? Yeah. And then it has 4 telomeres, each at the tip. And each tip has a telomere. So we have one chromosome, okay, possesses 4 telomeres. Okay, yeah, because this is one chromosome. 2 sister chromatids joined by centromere, but each of them has telomeres at the tip. So we have 4 telomeres. 1, 2, 3, 4. So therefore, if one chromosome gives us 4 telomeres, 32 chromosomes will give us how many telomeres? 32. Give us how many? You just said 32 times 4. So pick up your calculator and say 32 times 4. What does it give us? So 32 times 4, of course, will give us 128, right? Yeah. Pause it. So that's 128. All right. The diagram shows the cell cycle. During which phase do chromosomes condense and become visible? Yeah, during which phase? All right. So which phase do chromosomes condense and become visible? That's the diagram. Okay. I pause the video and try it. When they condense and become visible, they condense and become visible. Pause the video at prophase. So prophase is part of mitosis. Okay. So prophase is the first stage of mitosis. So in which part of the diagram do we have mitosis? If this is cytokinesis, next to it is mitosis. Right? This is mitosis. Okay. This is G1. A is G1. B is S phase, where semi-consecutive replication occurs. C is G2. Right? Yeah. So mitosis is D. That's where prophase is. The first there is prophase, followed by metaphase, PMAT, anaphase, okay, telophase. So don't forget this is metaphase, right? This is M, okay? So chromosomes condense at prophase, which is, so D is mitosis. Alright. Which row shows the correct number of each component of a single chromatid during anaphase of mitosis. Okay, so remember that during anaphase, pause the video and try it. Alright, yeah, this chromosome was like this. Okay, just pause the video and try it. Align at the equator at metaphase, but at anaphase it has changed. Anaphase, they are separated. So you see that at anaphase the chromosomes are already moving to the opposite pole. You can see that. So the sister chromatids are moving with their centromere leading. They are moving. They are pulled. They are being pulled apart. Right? Yeah. So the question now says, which role shows the correct number of each component of a single chromatid? Now this is a single chromatid, right? During anaphase, this is a single chromatid. So now A says how many centromeres? He has only one centromere. So A and B. Okay, so you see that C and D eliminated because they show two. Polynucleotide strand. Now you see one chromatid has one DNA molecule, and one DNA molecule has two strands. Okay, so one DNA molecule has two strands. So you see DNA molecule is double stranded, remember? That's double helix. And a chromatid has just one DNA molecule. Is that okay? So that means it's going to have two strands. Alright, so that's why A is the answer. A shows two strands. It's going to have two telomeres. You can see telomeres. One chromatid has two telomeres because one chromatid has two tips. So telomeres are the two ends. So A is the answer there. Okay, let's run. Some processes are listed: cytokinesis, differentiation, DNA replication, mitosis. How many of the listed processes occur during tissue repair by stem cell? Okay, by stem cells. Pause the video and try. Remember that when stem cells repair tissues, they have to first of all divide by mitosis. Okay, but before mitosis occurs, mitosis produces genetically identical daughter cells. So DNA replication ensures that genetically identical daughter cells are formed. So it occurs during that, right? So after DNA replication, after mitosis, the nucleus has split. The cell has to split too. So cytokinesis is there. When the cell splits, it's got to form a tissue. To form a particular tissue, the cell has to differentiate. Okay, so the four processes occur there. All right. The photomicrograph shows cells in various stages of cell cycle. Yeah, see, cute picture. All right. So let's see the question. Which stage of mitosis is not shown? Okay, I want you to pinpoint it. Which one is not shown? All right. Yeah. Pause the video and try it. Now this is prophase. How do I know? It's condensing. Can you see? Yeah, the chromatins are condensing. Okay. Now this is, can see, this is telophase. How do I know? The chromatids have separated, have reached the poles. So this is telophase. So telophase is shown here. This is anaphase. They are separating. Anaphase, okay? Separating. And of course, this shows us interphase. This is interphase, right? Nothing is happening, of course. You can see that interphase. Interphase is not even part of mitosis. So the one that is missing here is metaphase. Because at metaphase, they align at the equator. That's what is missing. Okay? Yeah, metaphase. Which description is correct? Yeah, pause the video and try. Which description is correct? Yeah, which description is correct? Let's look at it. Which description is correct? A says, pause the video and try it. A centromere holds two chromatids together until the end of prophase and attaches to the microtubules of the spindle. Okay, it does not hold it until the end of prophase. It holds it until the end of the cell of anaphase. Okay, Now, of course, until the end of, okay, you can say metaphase, because after metaphase, the centromere splits, okay, and then cytochromatin separate at anaphase. So it's not until the end of prophase. So that this is wrong, okay. B says a chromatid is one of the two identical parts of a chromosome and is made of proteins and two DNA molecules. Now, a chromatid is not made up of two DNA molecules, it's made up of one DNA molecule and 8 histone protein. One DNA molecule, okay? So it's not two, so this is wrong. Now, C says a chromosome is a structure with two identical parts made of DNA and protein found in the nucleus of a prokaryotic cells. Now, prokaryotic cells do not have chromosomes because they don't have histone protein. They don't have chromosomes, they have just DNA, naked DNA. So this is wrong. So we have D left. A telomere is a sequence of DNA nucleotides, yes, such as GGTTA, repeated many times, yes, and found at the ends of each chromatid, yes. That's a telomere. So if this is a chromatid, telomeres are found at the end, both end of the tip, okay, and they prevent loss of gene, okay. So this is it. This is the answer there. This is the answer. Okay. During metaphase, a scientist stains chromosomes of a diploid animal cell. You can see with a fluorescent dye to allow telomeres to be observed. This is the third time this question is coming out. Now this is metaphase, right? So we still have one nucleus. This cell has 26 chromosomes. So pause this video. How many telomeres would the scientists observe? You can see that this question is coming out all the time, like 3 times now. Okay. So if you ask 26 chromosomes, remember that one chromosome, right, gives us 4 telomeres. I've shown you that twice in this video. Do you see want me to show you that? Now at anaphase, chromosomes appear like this. They still appear as 2 sister chromatids held by centromere. Okay? And of course, each sister chromatids have 2 telomeres. So a chromosome consists of 2 sister chromatids. It's gonna be made up of 4 telomeres. So if one chromosome, which is this, one chromosome is this, the entire thing here, gives us 4 telomeres, then 26 will be what? 26 times 4. So 26 times 4 will give us how many telomeres? Okay? Pick your calculator and try it. 26 times 4, guys, according to my calculator here, is 104 telomeres. Delta is the answer. Okay. D. The photomicrograph shows cells in various stages of the cell cycle. Yeah, that's it. Okay, let's see. Which cells contain twice as many DNA molecules as a cell from the same organism that has just a complete mitotic cell cycle ending with cytokinesis? So which one has double? Okay, which one has double? Remember that. Look at it, guys. Pause the video and try it. It is a cell cycle, right? Pause the video and try it. This is mitosis, right? Which has prophase, metaphase, anaphase, okay, telophase, right? And then this is cytokinesis and a C, and then we have G1, that's part of the first part of interphase, and then we have S synthesis phase where DNA is doubled. This G2, so DNA is doubled. All right, so if I have after cytokinesis, this is 2N here, right? That's the number of chromosomes. This is going to be 2N here, but it's going to be doubled at the end of this S phase. It's going to become 4N. Okay, so G is doubled. Now throughout mitosis, it will remain 4N. Okay, now at the beginning of cytokinesis, it's still 4N, though by the end of cytokinesis, it will reduce it to 2N. So all of these stages, they are in mitosis, so they will all have double 4N, because they're asking us which cell contains twice. Twice means double as many DNA molecules. Okay, so all of these, they are all mitosis, so they are all doubled. So the answer is A, all of them. Okay. Uncontrolled cell division can result in the formation of a tumor. Which part of the cell cycle would take less time during formation of tumor? Which part? Okay, which part? So pause the video and try it. Remember that the normal cell has more of cytokinesis, right? So normal cells, this is mitosis. I've always drawn this. This is, of course, pause the video and try it. Okay, now the rest, they're interface. Okay, but when the cell becomes cancerous, the game changes. The cell spends most of its time dividing. Okay, so mitosis takes most of the time. Okay, uncontrolled mitosis, and of course cytokinesis. Okay, so you see that the interface has reduced. So this interface has reduced, so the interface is shortened. So that's the one that shortened interface. Okay, interface is shortened, or that increases. Interface is shortened, so that's B. The diagram shows part of the organism of a section of a DNA molecule and the associated histone P are in prophase of mitosis. Okay, that's it. Cute structure, very sharp. Which statement about the features labeled P, Q, and R during prophase of mitosis is correct? So I want you to pause the video and try it. Okay, thank you for hanging on. All right, yeah, so let's go, guys. In the diagram shows part of the organization of a section of DNA molecule and associated histone P, R. Okay, you can see that. All right, they say which statement about the features labeled P, Q, R during prophase. Remember, it's during prophase of mitosis. Please always read the question well. Now A says the coiled DNA molecule forms Q. Okay, it says forms Q. The coiled DNA molecule forms Q. Now this Q is actually DNA. Is DNA. Okay, and then wraps around histone protein R. Okay, to form small clusters held in place by histone protein P. No, histone protein is part of the folding. All right, so A is wrong. Okay, now B says the groups of histones P and its associated DNA. Okay, yeah, these are histones. All right, say DNA Q move close together as the chromosomes condenses around R. The chromosome, this is not yet chromosome. What I mean is that this is DNA folding around histone protein, and it's going to form chromatin. Okay, it's going to form chromatin. So it's not yet. Of course, can't use the word chromosome. Of course, it's going to become chromosome, right? But you see, it doesn't condense around R, and that's another thing you have to note also. Okay. Yeah, because R is part of the folding. So B is wrong. C says the histone P and R, yeah, they are both histone P and R, okay? Are made of protein around which DNA molecule? Q. So that's DNA molecule Q. Wrapped so that DNA molecule can fit inside the nucleus. Now this is correct, but that doesn't happen during prophase. Remember that we're looking at what happens during prophase, okay? Now that's why it's wrong. This says the linked groups of histone P and R. So P and R are linked groups and associated DNA Q. So remember, Q is DNA, R and P are histone linked group, form strands that fold and twist together to form chromatin. Remember that chromatin is formed during prophase. At prophase, the chromosomes condense. Of course, the chromatin condenses to form chromatids. Okay, and that's what happens at prophase. So D is the answer. All right. So how many copies of each DNA molecules will be found in a cell at the start of the stages of the mitotic cell cycle? All right. So how many? Okay. How many? Remember at the start. Remember what I showed you before. Okay. We had this. This is mitosis. M, and this is cytokinesis. So we're focusing on only G1. Now this is G1, right? So let's just focus. Of course, this is S. Yeah, this is S, and this is G2. G2, guys. So if you check here, you see that this is gonna be 2N at the beginning. Okay, that is right. So therefore we have only one copy of DNA here. This is chromosome, so one copy of DNA for each of them, right? Yeah, here we also have 2N, which is also one copy of DNA for each chromatid, but you know it doubles at S phase. So this is going to be, all right, doubled 4N. So that means we're going to have 2 copies of chromosomes right here, right? That, sorry, 2 copies of DNA. Yeah, so this also remains doubled 4N, right? 2 copies of DNA because it's been doubled. Now this is a C phase. So at the beginning of cytokinesis, which is C, we also have 4N, which is 2 copies at the beginning, right? But this is the end. Once it comes an end, it's no longer 2 copies. It's now 1 copy. You know, that's G. Starts all over again. So G phase, we have how many copies of each DNA molecule? Okay, of course, going to have 1. So A and B is selected. Cytokinesis, at the beginning of cytokinesis, we have 2. All right, but at the end we have 1. Okay, by that time the cell has split. So the answer is B. All right, thank you guys for watching. All right, see you next class on nucleic acid. All right, this is Shedro. Bye. See you next class.